You're watching CTOM TV. Knowledge is power.
There's also another wonderful tool that is available, and it's called RNA expression testing. And so basically what RNA is we look at something called messenger RNA. So there's a process where the DNA of a gene gets converted into messenger RNA, and that messenger RNA then gets transported to an area of the cell that converts it into its protein. And a protein is just basically a general term for the final product of a gene. So a gene is just the DNA, and the gene gets turned into a product. So the ER gene, for example, the estrogen receptor gene, is just DNA. And what happens is messenger RNA gets produced from that DNA, and it's estrogen receptor RNA. And then that gets converted into the estrogen receptor, the actual product, which is the estrogen receptor.
So can I interrupt you here? So if there is a mutation in one of those genes, that's why I'm going to have that overabundance of those receptors, or that's why things aren't going to bind properly. That ligand you were talking about with that cell surface receptor, is that how those mutations are going?
Yes.
And knowing that, if I can just keep trucking so that my thoughts are clear, and knowing which of those mutations are making things go sideways for me means that I don't have to settle for chemotherapy, perhaps, or
right.
I can actually try one of these. I don't know if you actually use the term targeted therapy here.
Yeah, precision oncology. These are targeted therapies.
Right.
We talk fulvestrant is a targeted therapy. CDK4/6 inhibitors are targeted therapies.
Yeah.
Because they target a specific gene pathway.
Right. So rather than having your oncologist just, hope for the best and put you on, say, immunotherapy or on one of these therapies, if you do the genetic testing, that tumor DNA and the RNA expression testing, then you'd know which treatments are actually going to work for your mutations.
Yeah. So I am kind of really simplifying things here. The RNA expression testing tells you how many copies of that gene are being produced. So, normally for a normal process, let's say you're only supposed to have 20,000 copies of a gene produced. Well, if that gene is overactivated, then it's going to produce 100,000 copies of that messenger RNA. So the two tests are the tumor DNA sequencing and the RNA expression testing. They're very complementary. Tumor DNA sequencing has been around since 2006, 2005, when we first sequenced the human genome. Expression testing has really just come of its own recently, and now we're able to look at all 20,000 or so human genes with a single expression test. So these particular tools are incredible for precision oncology and for really, determining what's best for a cancer patient, not only initially, but all the way through their treatment and through the course of the disease as the disease changes. So alterations, we call them molecular alterations, can either be mutations. And what that means is they can break off the receptor, they can cause that particular gene to stay turned on, or they can cause that gene to actually turn on cancer-causing genes. So it's kind of like a cascade. So you're changing the shape of the key of the lock, so it's going to fit different locks. And some of those locks, all of a sudden, may be cancer-causing genes. And so expression testing doesn't really look at the mutations. It tells you how much of that gene product is there. And that's also very important, because let's say you do genetic testing and you don't find any mutations on your HER2 gene, or you don't find any mutations on your CDK4/6. There can also be other things called epigenetic pathways that cause an overexpression. So think of mutations as the function of the gene, and think of expression testing as the turning on and off of the gene. So completely two different things. So if you were to think of the wiring of a light switch going to a light, think of mutations as damage to the wiring inside the wall or damage to the light switch, and think of the expression testing is actually turning that gene on or off, and turning it on for longer than it's supposed to be turned on is something that is very important. You want to know that. So both of these tests, expression testing and tumor DNA sequencing, are very important for every cancer patient.
All
They
right.
allow us to determine all kinds of different things.
Thank you for watching CTOME TV. Subscribe below and stay informed.